This is our project on the regeneration properties of traumatic and non-traumatic injuries inflicted on members of the brittle star species. Our experiment was to determine the requirements and elements of regeneration of organisms who have regenerative capabilities such as starfish or the brittle star. Our independent variables for the experiment were traumatic and non-traumatic injuries, which were used to determine whether the brittle star will be able to regenerate or if it sustained too much damage.
Our experimental procedure consisted of a traumatic procedure for when a traumatic injury is inflicted on a brittle star, and a non-traumatic procedure for when a non-traumatic injury is inflicted on the brittle star. The beginning of the traumatic procedure involves picking out a large brittle star and using tension to apply a traumatic injury, gradually applying more tension until the arm separates. It is important to ensure that autotomy does not occur in the brittle star, which is when it chooses to separate its own limb from its body, which results in a non-traumatic injury. It increases its chance of survival, and the traumatic injury is made along a line that does not allow autotomy to happen. After getting the traumatic sample, it is put on a slide, and we can analyze it under a centrifuge or under a microscope rather. And the procedure for the non-traumatic injury is the same, except autotomy is allowed for the brittle star. So self-separation of its own limb is what we want in this situation, and then we can analyze that same sample under a slide and compare the cells and look at the different communities. It's also important to note that while non-physical means of limb separation is possible because of chemical instability in the tank, these samples are considered non-traumatic if autotomy is produced from unstable chemical standards in the tank. However, these samples were not used in our data set. We used PCR and electrophoresis procedures to analyze tissue samples that were collected, in addition to the microscopy that we did for the cell communities from each sample. The PCR tests consisted of using a DNA ladder that we compared in the 2 kilobase and 6 kilobase series for a traumatic sample. However, the non-traumatic sample is yet to be analyzed.
We wore gloves in order to prevent any contamination with the samples or the organisms or the tank water. But however, there were no dangerous biological chemicals or properties being used in the procedure itself.
Test chemicals were handled according to the safety data sheets. Our observations on the non-traumatic slide observations included plentiful cell populations, especially near the coelomic cavity, which is the circulatory system of the brittle star. So any thriving cell communities in that area inferred that the brittle star was progressing with its regenerative properties and was about to form a blastema. And after a week of observing, the injury appeared to be fully healed and the arm was lengthening. However, in the traumatic slide, we observed opaque cells and very short cell communities. The time period that we observed the traumatic samples, or the starfish that we collected traumatic samples from, they seemed to have little to no progress in regrowth, and it seemed the wound had just been healed over and then progression of regeneration has stopped completely. So the brittle stars were more inclined to regenerate when their survival chances were favorable, and traumatic injury and traumatic recovery, where the limb does not regrow and the brittle star just maintains its primary life functions. It seems that this is the case whenever it's not favorable to use more energy to regrow that limb. The traumatic injuries were a survival mechanism, that their stop of regrowth is favorable for brittle star survival. Thank you.